So basically, this is a big emphasis in my lab now, is trying to use proteomic technologies again to find novel immunotherapeutic targets. And as we know, BCMA-targeted therapies have shown so much promise, whether CAR T cells or bispecific antibodies that are being presented at the meeting here, as well as antibody-drug conjugate we have therapeutically available, as well as monoclonal antibodies targeting CD38, for example, SLAM-F7 that we have therapeutically available. So these are all cell surface antigens that we know can have therapeutic promise. But the question is, unfortunately, myeloma still remains incurable. Our goal is to get to cure, of course, but right now patients still relapse. Even after BCMA-directed CAR T, patients still relapse, even if it's 2 years down the line. What do we do for those patients? What can we offer them? So our goal is thinking, can we still profile the surface proteome of these myeloma plasma cells to find new targets that maybe we could use in these refractory settings? And so what we did in our most recent study, though, is just published. We profiled the cell surface proteome of several different multiple myeloma models, as well as primary myeloma patient samples. We basically found several hundred cell surface protein antigens that were present. We could quantify their abundance in a relatively unbiased fashion. And so in this context, we found and we developed a scoring system to say what are targets that are selectively on plasma cells and not on normal cells that you don't want to hit that would cause off-target toxicity. In our ranking system, BCMA came out #1, which was very reassuring because we know is such a promising target in myeloma. But we also found several others we thought were quite interesting. And one of the ones in particular that we felt was particularly promising is called CCR10. This is a chemokine receptor, hasn't really been identified on plasma cells before, but we profiled many primary plasma cell samples, saw it was uniformly highly expressed on myeloma tumors, is not present on normal peripheral blood mononuclear cells, and we were even able to develop a proof of principle CAR T cell versus CCR10 that we're still working on further developing in our lab. And so I think CCR10 in particular came out of the study as being very promising. But in addition to, we were able to find biomarkers of resistance to first-line therapies. We could profile the cell surface proteome before and after bortezomib or before and after lenalidomide, both chronic resistance and acute drug treatment. And so I think by looking at the shifts in the cell surface proteome, we could come up with new therapeutic markers that are specific for resistant samples, new diagnostic markers that could be relevant to have early diagnosis of resistance to therapies. And so this is really the promise. And I think what we want to do going forward is do this more routinely, particularly for patients on immunotherapies, and trying to say how are their tumor cells changing in the cell surface antigen profile after CAR T therapy, after bispecific therapy that could suggest those second-line therapies for patients who do become resistant. So that's really where we want to go in the future. We think the cell surface proteomics is very powerful for that.